As we've seen in previous videos, amino acids are compounds that contain a amine functional group and a carboxylic acid functional group. And they're separated by what's known as the alpha carbon, which has a unique functionality known as the side chain or the R group. When these amino acids condense together, they form an amide linkage, or in the context of biochemistry or biology, we know it as a peptide bond. Okay, so this bond right here is the amide linkage or our peptide bond. Okay, so what's really important to recognize in terms of peptides is that because the C terminus and the N terminus of individual amino acids are tied up in this peptide bond, they're no longer reactive. So as longer peptides form, the only thing that really dictates the functionality or the physical or chemical properties of amino acids are the R groups, the side chains. So what we're going to be doing in this unit is exploring these side chains and exploring the chemistry that these side chains can undergo and how the side chains can influence the chemistry and the physical properties of different peptides. So you should have been able to find a table of the common amino acids, and that shows the structure of the side chains as well as the pKa of the amino group, the carboxylic acid, and the side chain if it can participate in acid-base chemistry. So what we're going to do in this video is explore the acidic and the basic amino acids and figure out what form they're most present in under biological conditions. So let's start off with thinking about the two acidic amino acids, aspartic acid and glutamic acid. Okay, as you see on that table, the three-letter codes for these are asp and glu. Okay, so if this is our peptide backbone, then aspartic acid has one carbon followed by this carboxylic acid functional group. Glutamic acid is two carbons followed by the same carboxylic acid functional group. So they both have this carboxylic acid on the side chain. Okay, the table tells us that asp has a pKa of 3.90 and glutamic acid has a pKa of 4.07. Okay, so if we think about this now in terms of what we learned about speciation, right? So if we are at a pKa or a pH of 3.9, then anytime we have a pH that's more acidic than this, then we are going to have the protonated form of our amino acid present. And anytime we're more basic, then we have the basic form that's present. Okay, so since we know that biological pH is up here at pH 7, this means that for both aspartic acid and glutamic acid, under physiological conditions, under biological pH, it's the basic form of these amino acids that are most prevalent. So putting this in the context of the structure, this is the acidic form, right, the protonated carboxylic acid. So our deprotonated form of aspartic acid, known as aspartate, has a negative charge. The same thing goes with glutamic acid. So glutamic acid, when it gets deprotonated, it now has a negative charge. So under physiological conditions, both glutamic acid and aspartic acid are anions. Okay, so let's take this same logic and apply it to the two amino acids that have really basic pKa's. So we have lysine and we have arginine. Okay, looking at this chart, lysine has 4 carbons, so 1, 2, 3, 4, and then this NH2 group. Okay, if you draw out the Lewis structure, NH2, this nitrogen has a lone pair, which ends up being reactive. Okay, arginine has 3 carbons followed by what's known as a guanidium group. So if this is the backbone, we have 1, 2, 3 carbons. We now have a nitrogen, another carbon attached to 2 more nitrogens. And one of these has a double bond. And we fill in our hydrogens here, and we fill in our lone pairs. So this is a fairly tough structure to remember. But what's really important to recognize is that both of these are drawn in their basic form. So in both cases, we have a lone pair that can reach out and grab onto a proton. Okay, so these are in their basic form. So the basic form is neutral. All right, let's think about our pH scale. These both have pKa's way up in the basic range. So lysine has a pKa of 10.54. Arginine has a pKa of 12.48. Right, so these are both way up in the basic area, meaning that under physiological conditions, right, way down here at pH 7, we have the protonated form that's most present. As we just established, the basic form is neutral, so the acidic form of these two amino acids are going to end up being protonated, so they have a positive charge. So lysine, 1, 2, 3, 4, NH3 plus. So this is the protonated form of lysine. And arginine, we have 1, 2, 3 carbons, nitrogen onto another carbon. So here we've protonated this nitrogen. So arginine, in its protonated form, its acidic form, also has a positive charge. So the big punchline here is these 4 amino acids are really common charged amino acids. There are ions under physiological conditions. Lysine and arginine are both cations. Glutamic acid and aspartic acid are both anions. So when we think about the chemical and physical properties of peptides, it's important to remember that these 4 amino acids will carry a charge, and that's going to influence what these peptides can do and what they can interact with.